Let's share the screen. All right, so here's our agenda that Jenny just went over. And so we're going to start with some going over the colors again. We ended it last time, but I want to make sure that we covered that kind of fully. So I'm going to start by opening this confocal stack. This is actually what we're going to use for almost the entire demo today. So this is cochlea, this is actin, and this is a stain that does some nuclear staining on outer hair cells. So we have green and red in this case, and currently they are in their own channels. So there's a lot of different things we could do with this. One thing we might want to do is see both channels at the same time. So if we go to Image, Color, and we have a lot of tools here, and many of these tools will kind of do the same thing. I'm going to use this Channel Tools first. This is a tool where we can first go and look at each channel. We go under this menu here. There's a lot of different options, including grayscale, color. We can do some inversion, stuff like that. But other things we can do is we could make a composite, which we're going to do in a minute. That will combine both color channels into the same channel so we can see them at the same time. We could also recolor any of the channels we want using this menu here. So right now I'm on channel 1, it's currently green. Let's say I wanted it to be blue for some reason, I could do that. I'll show you a more realistic scenario that you should consider doing for publication. So currently our channel 2 is red. Some people are red-green colorblind, and I can kind of attest. I've had a trainee in the past who was red-green colorblind. It's really hard for these people to kind of see these images, but there's an easy way to fix this. So instead of red, we can make this magenta. And this is pretty common. Some journals will even ask you to do this. I'm not sure how many, but it's happened in the past to me. So now we have channel 2 as magenta, channel 1 is green. If we want to see them both at the same time, we can click this Make Composite, and then we can see them both at the same time. So that's one way of doing this, but let's go over another. So I'm going to close this, and I'm actually going to close this as well and just reopen it. So another way is we could start by splitting channels. So image, color, split channels. This will just take each color channel and make it its own either image or stack depending on what you're starting with. We have stacks, so they're going to split as stacks too. So now we could individually manipulate these channels, you know, do the brightness contrast like we discussed last time. Do anything else we might want to do, and then when we decide we want to recombine them, we can go to Merge Channels and we can pick. So right now it has C1 as red, which would not be correct in this case, and C2 is green. So we're going to set this C1 as green, and we'll probably do another magenta for C2. And I always check these boxes, keep source images. It just won't delete these in case you missed something, and ignore the red-green that's in there now. So you get this image again. Now, a word of caution about the magenta. It's really good for publication, but if we want to split this channel, it might turn into both our red, or a magenta might go into both a red and a blue channel, which is sort of annoying to deal with. So this is something you typically want to do towards the end of your analysis. One more thing. I'm just going to open it one more time. So image, color. This one I'm just going to directly click, make it composite. That'll combine them. They're combined again. And let's say we have an image and we just want to save it so any computer will open it and we'll just see this. And then we could go to Type, RGB Color. So right now It's 16-bit. That means everything's still stored in grayscale, but the computer knows to apply a lookup table, in this case red and green, to each channel. We burn this into RGB color. This image now, the pixels are actually colored red and green, and any computer will open this and recognize it as a color image. But you've lost a lot of information here, so that's the downside. It usually increases the file size quite a bit too. Now let's say you've already burned in an image, but you realize, hey, maybe I really do need this green magenta. There are tools for this too, and they're useful for looking at publication figures too. So if we go again to color, and down here is a whole bunch of tools to deal with color blindness. So here we have replace red with magenta. Click that, and it does that. This only works on RGB images though. It will not work on the 16-bit. It won't work on a stack. And it will even take this out of a clipboard. So if you cut an image out of a PDF and you click this in FIJI, it will automatically convert it. All right, I think that is it for colors from last week. Are there any questions about colors?
Please feel free to type in the chat or unmute yourself and ask a question if you'd like.
Well, if there are no questions, we'll just move on to stacks. So I'm just routinely closing and reopening the same file just to make sure that we're starting with a clean file each time. So here's our file. So this is a stack. It has two colors. Here's green, red, and then we're seeing the planes through the image. So these stacks are really cool, but you often will need to kind of reduce their dimensionality so that you can actually do something with them. So I'm going to show you some basics on how to do that now, and then I think the next one we're going to do some more complicated, like in time series, how to display it. I'm going to show you some of the more simple things that are really commonly done. So the first one, I'm actually going to do a color modification quick. I'm just going to make this a composite. The first one that's really commonly done is Z projection. So this is collapsing our Z plane into a single 2D image. And the reason you might want to do this is it makes it easier to display, and it generally will make a really pretty image. So how to do that? Go to Image, Stacks, and we have Z project. If we click this, these are the slices where it will project from. Sometimes you don't want to project through the entire stack. If you have a region of interest, we can just project through that part. And this is a fairly small stack, and we'll just leave it the full amount. Now there's many different types of projection type. The most common one is probably max intensity. The theory behind this is that your brightest pixels are your pixels in focus, and so you want to see those through the projection. For electron microscopy, we often use some slices as well, so that can be potentially useful. If you are looking at some sort of inversion, minimum intensity can be equivalent to maximum intensity. So we're going to do maximum intensity for this one and just hit OK. And it's going to give us an image like this. So what we're seeing in this case is the brightest pixels in X and Y. Well, in each XY plane, where the brightest pixel in Z was, that's what it's showing. So here we can see a bunch of stereocilia projections at the top, which we would have a lot of difficulty actually seeing, except for in a couple frames in the stack. Let's go on to orthogonal views. So we can also look at multiple planes at once, not just the kind of XY plane going through it in the Z plane, but we can look from the other planes too, the XZ and YZ planes. So we go to Image, Stacks, Orthogonal Views. It'll open up two other windows on the bottom and the right of the image. And wherever we place this X, we can drag it around. It will show you the other two planes. So if I put this at the top of this cell, here we can see the, let's see, this is the YZ plane. So the top and then going down into the nucleus, and this is the other plane going the other way. And we can drag this kind of crosshair in any of these windows, and the other windows will adjust to the same location. So it's a good way of figuring out where you are in an image, and it's also a good way to actually evaluate the quality of the image itself. So typically your XY plane will look quite nice, but your Z plane, depending on a lot of different factors, may not look as nice. So here you can do things like check the alignment and make sure that it's all making sense. All right, and if you want to get out of this, it's really easy. Just close one of these two windows and it just closes on its own. All right, the next one is stack to image. So if we want to collapse our stack into single images, there's a couple ways of doing it. The simplest way is to go stack to images, and it will literally take every slice here and put it as its own image. I'm not going to do that right now because we have a lot of the stack is big enough where it'll be annoying. Instead, I'm going to show you another way. So if we go to duplicate, we can duplicate any part of this stack. So we can do one color channel, two color channels, any Z planes we want. So for example, if I only want planes 10 through 12, both colors, I could duplicate that. That'll look like this. And then if I wanted to, I'll show you the other way now that it's smaller. We can go to stack to images. So now each plane, and I think each channel is its own image. Now vice versa, if for some reason you wanted to import your Z plane as single images and put it to a stack, the reverse is possible. You would open them all and you would go images to stack. Now if you do this, you have to be careful because it will take every single image that's currently open and try and make a stack with it. And so if you have images open in FIJI that aren't related to your stack, they will end up in there. So it's best to just start with a clean, just open FIJI, open the files you need and do that. All right, are there any questions on this part of stacks?
I have a question about that probably other people do. Is there a way to go images to stack and just point to a folder?
If you were to do that, I would do it a different way. I would probably go file, import, image sequence.
Yeah, that's what I was asking.
Yeah, and yeah, we can actually do that.
Images to stack.
We did that last time, I think.
I think so.
Oh, this is the old one. Types of files, interlace TIFFs. Well, anyway, that's probably the easier way of doing it. Any other questions? If not, we'll move on to 3D viewing. So I'm going to show two different 3D viewers that are in FIJI. They're a bit different. The first one is the one that I've used a lot in the past, and the second one is a bit newer and potentially can handle bigger files a little bit better. So the first one, if we go to plugins, we have just 3D viewer. And click that and get a message from the console. And then it's going to open this window here. So it says, what image are you wanting to open? What name do you want to give it? How do you want to display it? We can do volume, orthoslice, which is sort of what we looked at before, surface, things like that. The resampling factor, I always change back to 1. This is essentially how much the image is binned. And then what color channels it's reading in. I'm going to uncheck the blue because there's no blue in this, this image. Click that. It will take a second to do some calculations on the 3D. And hopefully, there it goes. All right, so let's see. This laptop may struggle with this a little bit. We'll see. So I'm trying to use the mouse wheel to scroll up just to zoom it a little bit. So we can do things here like rotate, reorient. We also have the ability to change the transparency of the image, either through thresholding or just directly changing the transparency. Either one. That's pretty computationally expensive, so I'm not going to do it right now. We also have the ability to change the background color. So right now it's straight black. I'm going to set it to be white, hopefully. So 255 in every channel. Let's see. It's almost there. It's a little intense for, this is probably something you want to do on a workstation. Well, I'm going to give it a second to catch up, and if it doesn't, we'll move on to the other volume viewer. We'll see what happens. So this guy is kind of busy. We will attempt to open the other one and see how it goes. So the other volume viewer likes RGB images. So I'm going to go to File, Type, convert this to RGB color. And we're going to wait. All right. Sorry about that.
It's a great example of why you should use the workstations when you're dealing with big data.
Yeah. Yeah, I should also mention that that particular plugin doesn't like large files. This file isn't particularly large. It's only 39 megabytes. But I tend to avoid putting anything larger than a gigabyte in it. It doesn't seem to handle it very well. So I'm just going to make this a composite again. RGB color. It's just asking me if I want to make everything RGB color, and I say yes. And we'll try the other one. So instead of 3D volume viewer, I'm going to go to just this one at the bottom, volume viewer. And this one seems to handle big data a little bit better. So it's opened it just in a slice right now, right through the middle. I'm going to set it right away to look at volume. And I'm going to change the background color from blue to white. And right here is scale. So right now this is at a scale 1, but I like to zoom it in a little bit to see what I'm looking at. So in this one, we can do a couple different things. So we can look at different planes just by clicking them. So XY is just your typical top-down plane. Here's your side planes. And we can also do arbitrary planes by kind of dragging these through. What else can we do? So background color, if you get really lost, you can hit this reset and it will just basically set everything back to normal. I use that all the time. There's several different interpolations. I don't know which would be best. They will largely look the same. You can also do some arbitrary like projections. So we can project at an angle, for example. This is a Z project, but through an angle through the sample. So not just from the XY plane, but from any arbitrary plane we want. All right, I'm gonna close this and I'm gonna shrink this file and try and open it again in the 3D viewer, just because there's a cool movie making feature in there. So to shrink it, I'm just gonna scale this. It's already a pretty small file, but we can shrink it by half and it'll be really small. We'll plug in, it's 3D viewer. And I'll leave the resampling on too as well, so it's even a little bit further binned. Well, yeah, this one just doesn't run on this computer, unfortunately. This one has the ability to make little movies that just rotate through the sample automatically. So if you're on a bigger computer, it's under View, and it will take like a 360 recording at any arbitrary axis you want. It makes that into a stack, and then you can either export the stack as itself or as a movie or as a GIF. And we can go over that on the next one. I'm going to just close FIJI again. All right, do we have any questions while we're waiting for FIJI?
There's one question in the chat. How can you save a 2D picture of the volume for publication?
Okay. That one we can do. So I'm just going to go through the same kind of stuff I was doing before, make a composite, RGB stack, keep source. We'll go back down to volume viewer, and volume. I'm going to change the background again to white. It's really up to you what you want that to be. And here we have this snapshot button. So snapshot will literally just take a picture of whatever you have here. We can use this to zoom out, or you can zoom in and hit snapshot, and you will get exactly that image. Now, I would probably, for publication, I might not necessarily use FIJI. This is more for like lab meeting stuff and just your general analysis of the data. There's some software packages that are probably going to handle this a little bit better. And I think we're going to be talking about those in the future. Any other questions?
Are there settings in there, though, for that snapshot that will give you different?
The settings are entirely determined here. So
oh, I see. They're down. Okay.
Let's see.
Any other questions on volume viewer?
Well, if not, we'll move on to quantification, because quantification can be pretty involved. All right, so I'm just going to open this guy again. We're going to go through some basic tools first, and then some more complicated things. So we can measure a lot of different things in FIJI. If we go to Analyze, Set Measurements, these are all the things we can measure within an ROI. We're going to go how to draw an ROI and how to save them in just a minute. But I know what most of these do, but not all of them. So in general, I recommend capturing a lot of information. You can always throw it out later. Area, standard deviation of the brightness of the pixels, min, max gray values, median gray value is in here somewhere. I often use shape descriptors too. This integrated density is taking the area. It's essentially a summation of your pixel brightness over an area that you select. You can do perimeter, centroid will find the center. Anyway, there's a lot of options here. You can pick them, hit OK, and then let's go over how to actually make an ROI. So the most simple ROI you could make is a square. That's with this rectangle tool here. So you can make a rectangle. If you hold Shift while you drag this, it'll be a square. Similarly, this oval tool will make arbitrary ovals. If you hold Shift, it's stuck as a circle. This is polygon tool, so you click, and every time you click, it makes a new vertex in the polygon until you connect it to the first vertex. This next one, which looks kind of like a kidney bean, this is a freehand tool. So this is one of the ones that I find I use a lot. It's kind of basic, but it works pretty well. Typically when you use it, you want to be really zoomed in so you can see individual pixels and kind of trace them accurately. Kind of like that. This align tool, we went over that last time for how to set scale bars and things. We also have a point tool, so this just puts a point in. You can save the position of that point. We'll go over that. And this one is potentially very useful. So this is sort of an automated tool. So if you click somewhere with it, it just makes a point initially. If you double click the picture, on the, I guess it's called the wand tool, we have this slider. And this slider will expand your point based on the brightness around it. So you can potentially use this to get a lot of areas much quicker than if you were to do it by hand. So I've set this to this number, which is tracing out the inside of this nucleus. Hit OK. And then if I click in other places, if I've done this right, it should do a similar job getting that ROI. So this is very dependent on your brightness being consistent. And it can pick bright things or dark things. It doesn't matter, but there has to be a boundary. If we get it too large, I will attempt to do that, it will probably travel out to the outer sides of the sample. So this is basically selecting the whole sample. So if you find you're using this tool and you just can't figure out what's going on, reduce this value and see if it gets back to small again. All right, so those are the basic how to select things. Now if we want to measure something, we can hit Control M, and that will do all the measurements that we just discussed. So these are the things that I selected, and so we have like the area in microns, the mean, the standard deviation, min, max, etc. However, I usually do this later. So the other thing that's really important for doing these kind of measurements is being able to basically save them all. And so for that, we need what's called the ROI Manager. It's under Edit, Selection, and if you just hit Add to Manager or Control T, this will come up. So this is the ROI Manager. And what it will allow you to do is add things and label them and save them. So if I wanted to save a bunch of these nuclei, I can click them. I'm hitting Control T, which is adding them over here. And if we show them with this box, Show All, we can see that I've selected all of these. Now, we can do a lot of different things here. I'll show you the simplest first, and that is if we just select them all and I hit Measure, whatever I've selected under the set measurements is going to come up. Now there's a bunch of things we can do here. One of them is rename, so we can rename this, for example, this outer hair cell 1, outer hair cell 2, etc. You could go through the whole thing. Other things we can do, if we hit this more here, a whole bunch of options come up. We're not going to use all these options, but we're going to definitely use a few. And one of the major ones is save. So this will save these ROIs that you've selected, because potentially it's a lot harder than just clicking these spots that I clicked. You might spend hours tracing things, and you want to be able to go back to it. So if you go to save, and what I would recommend is using the exact same name as the file, and then it has to have that .roi in it. So this would be cochlea. And that's so you know that this ROI is associated with that file, because FIJI does not know or care. Likewise, if you have the file open, you can open this ROI manager with Control-T or just through the selection, and then hit open, and you can select the ROIs. Now I should mention, this is very, very important. When you're saving ROIs, you have to have them all selected if you want them all saved. So click, I'm holding Shift just to get all of them, and then you would do the save. Don't complain that I already did it. If you just have one thing selected, it's just going to save that one thing. So that could potentially be disastrous. Let's see, what else? So the other thing that can be really useful is these logical operators. So let's start with the OR. So if I wanted these things, let's say these 3 ROIs to be the same ROI, I could go there and I could go more, and then OR, combine, and they should have become a single one. I don't know if they did or not. Let's see. Oh, and then I need to hit add, and now it's a single one. Alternatively, if you want to know or select things based on some area, like for example, if you're segmenting something in a cell or a specific region, like let's say I just want things in this region defined by this box, so these ROIs defined in this box, then I would, well, let's make the box an ROI first. It doesn't touch 3. So I'd add that. We'll just call this region of interest. And so then I could actually select everything here and do an AND function and add that. Oh, image does not have an active selection. Let's see. Well, it should combine them. The other things that you could do, if you have a bunch of regions that are combined together, you can split them. So that's split. It will do its best to split ROIs that are in the same category but are not touching apart to their own unique position. This is a not operator, basically. So something that's not in this region, you could define. So that's potentially useful if you want something not in a specific cell type, for example. Okay, let's see. This flatten button here allows you to produce an image with these ROIs on top of it. I'm getting a warning here that there's a big stack here, and so we probably just want to look at this on just the single image. Oop, too big. Let's shrink that down. And so this would, well, we can just do it. This will flatten this so you can save it, just to show what you've done. This isn't for publication, it's more for just showing what you've done somewhere. I think that's largely it for the ROI manager. So are there any questions on this? Guess no questions. Okay.
No questions in the chat? Anyone?
Well, we have some time left, so I'm actually going to get to the other things I wanted to go through. So we can also do some kind of spatial frequency things in FIJI. This can be a way to detect repeating patterns or find out how close things are to one another. I'm just going to discard all this. Let me show you two ways of doing this. So here we have a bunch of microvilli, and potentially you might want to know what's the distance between these microvilli. So there's a couple ways that I've done this in the past. I will show you both of them. So I'm just going to duplicate this first, and I'm going to get rid of the information bar at the bottom because we don't need it. And then there's a function here in process called find maxima. So this will place points at the brightest pixels in something. If we do it right now, it's not going to work very well. If I click it, I always click preview point selection, and it's going to try and calculate points. There's like a million, way too many. But we can make it a lot easier for it to calculate this by essentially applying a filter. So we have a bunch of filters here. You need to be very careful with filters when you use them, and always report that you did use them. In this case, I'm going to use the Gaussian blur. Hit preview. This basically is similar to a low-pass filter. It averages nearby pixels. At a very low value, like 2, it actually makes the image look better in SEM usually. But we want to take this kind of to an extreme. So this is averaging 20 pixels around a pixel. In that case, we're kind of making like points here. Hit OK on that. And now if I go to this find maxima, it shouldn't be as ridiculous. So it's found a bunch of points. They're pretty good from what I can see, but they're not exactly right. So up here, this prominence is sort of its criteria for selecting them. So a higher number typically results in less points, so it's more strict, and a lower number is we'll find more points based on, I guess, closer proximity to the maxima. So what number to pick is entirely up to your sample. You just see what looks correct. So in this case, 8 is pretty good. There's a couple that are missing. You could go and add those yourself using the point tool. I'm just going to hit OK on that. Oh, actually, you have to leave it open. That's right. Oh, no, you don't. OK. So now I have all these points. What do I do with them? So you can go File, Save As, X, Y coordinates. And this is basically just a list of the coordinates of all these different points, and you can open it in Excel, or I usually use MATLAB to do this kind of analysis further down. I'll show you another way to do this too, where we can actually just use FIJI. So I'm going to close that. It's probably going to ask about the points. So another way of doing this is by Fourier analysis. So Fourier analysis is looking at spatial frequencies. And this only really works on things with very regular patterns. These microvilli, I suspect, are regular enough to work. So if I go to Process, FFT, I want to have a square ROI for this, and definitely not any text. Text has lots of repeating patterns in it. It will definitely show up, so make sure that's out of it. calculate the Fourier transform, and this is what it looks like. So Fourier space is reciprocal to normal space, meaning that things that are close to the center are actually low spatial frequencies, and things that are further out are higher. Basically, pixels here represent frequencies that are repeated very commonly. So we can look at these. If I just take a tool and drag it around, if I go to this point, so if we look here, I can't point to this thing at the same time. If you look on the FIJI menu, just below File in the rectangle tool, you'll see an R. R equals 0.1. So that's the spatial frequency, essentially 100 nanometers. And as we go out, it'll go down. And each of these represents a spatial frequency common to this image. So actually, these spatial frequencies in the center represent the actual microvilli, and these other ones are the gaps between microvilli. So it's a kind of quick way of quantifying things that have very, very consistent features. All right, do we have any questions on any of this?
There's one question that popped in right before you started the spatial stuff back to the ROI manager.
Okay.
How can you segment a cell for quantifying the area of vesicles inside the cell using the ROI manager?
That's a good question, and something we actually do fairly frequently. So I don't have a great example image for that. I'm just going to open our cochlea image again.
That's a good point. Quantifying vesicles inside a cell for area can be extrapolated to quantifying something inside of something else, right? Cell, nucleus, vesicles, those can be whatever those things are. So this would apply to other things.
Yeah, so how I would do this, I would, let's get our ROI manager open. So the first thing I would do is trace out the entire area of the cell. I don't have a good example of just a single cell, so I'm going to do this whole strip of cochlea instead. Add that. And then this is where naming is going to become super important. So we'll just call this the cell area. And then you're going to go through and pick all your vesicles. We'll just call these vesicles for the purposes of this. And ideally you want to rename all of these too, but it would be a lot of work. Then, what you're going to do, if you're just interested in area, we can get rid of all these other things. And we're going to measure the area of this whole thing. And then we can go and we can measure the area of these things. And then you just take this in Excel and you can do your math there. So you have your total area of your cell, you have the total, all the little areas of your vesicles. You can sum them together if that's what you're after. We could potentially do a union here, so do an OR, and that would be all of these and measure them as a total unit as well. It's kind of an extra step that probably isn't needed. You can just do it in whatever graphing program you use. Sometimes people will also remove the area of the nucleus depending on what you're looking at, so that might be a third thing you would segment.
Any other questions about the ROI manager or about the spatial information? Thanks, Bill. That's a question. You're welcome.
I have one other thing that someone asked me beforehand to show, so I can do that because we have a few minutes, I think. The question was kind of an interesting one that I hadn't heard before. It was how to add a single overlay to a stack. So in this case, we have a confocal stack here, and they want to have this single image over the whole stack. So this took some thinking. It is not done through the stack menu, which you might think. Instead, we do it through the image calculator here. So process image calculator. And then we need to select the correct images. This is the stack. This is the single image. And then we'll create a new window and a 32-bit float just to be careful on top. And it will say, do we want to do this to the whole stack? We go yes. And we end up, nope, maybe we don't check that float. Hold on. Image calculator. We lost our color information. There it is. It's kind of the stack floating over this image. It creates kind of a cool thing. So that's all I have. I'm happy to answer some more questions. Next time we'll be covering time series data and how to present it, and also some ways of presenting stack and 3D data just in PowerPoint and in papers.
Okay, I do have an extra question about the segmentation. So when you do the segmentation in black and white images, you can also get the intensity measurements.
Yes.
In that segmentation, right? So it's not just for, I don't know, electron microscope, for area, but also for intensity, right?
Yeah. So here, I'll bring the screen back up here. Yes, so for electron microscopy, it will just read the brightness value of the pixel. If you're doing TEM, you're actually looking for dark things, usually not bright, although you could invert it if you were interested in that. I don't have any TEM examples for this one. But yeah, the FIJI doesn't necessarily care too much about pixel values. To it, it's always just gray values, even though it looks like it's green or red. To FIJI, it's just reading the actual intensity. So to read intensities, that would be typically min, max, standard deviation, mean. If you have a single point, it'll read the brightness of a single point. Typically, you want to average it over a couple points, though, or a large area, depending on what you're looking at. All right.